So liver is the only internal organ which can regenerate itself even if you take out 70% of it. So if you're left with even 25 to 30% of the liver, it has the ability to grow back. Patients with liver, they usually end up in hospitals at end stage when you don't have any kind of cure or no therapy. So it's not possible that a single organoid can do completely say it's the whole organ. No.
Hello and welcome to the 3DCC platform Conversations, wherein we bring the voices who are working in the field of healthcare to build India's future and beyond. And today I'm very pleased to be joined by Dr. Savneet Kaur, Associate Professor at the Institute of Liver and Biliary Sciences. Her research focuses particularly on the vascular niche in the liver biology, and also her team has been developing the liver specific bioscaffolds that support the long term hepatocytes cultures and enable the advanced drug discovery and testing. And she also works extensively with the liver organoids for the disease modeling and translation applications. Also, Dr. Savneet, it's a pleasure to have you over here first. And her work is immensely about the organoids, 3D cell culture in the field of healthcare. So we will be having the conversation regarding the same. Welcome you here on the podcast.
Thank you so much.
So before we begin with the whole scientific conversation, I would want to know how your journey in this whole domain has been so far for you.
Talking about the profession, so I am a PhD in biochemistry from the Institute of Genomics and Integrative Biology. So actually when I joined PhD, it was all about working in the lab for hours, day and night, and thinking of new results. And we were working something, the problem which was given to us by our mentor, the supervisor. I think after doing PhD, that approach of just working in the lab changed into thinking.
Okay.
So it's like a transition. PhD is all about learning, but then during learning, you also learn to think. So I think after that, we realized that it's just not the skills, the technical skills, it's also about the questions you ask.
Yeah.
Because it's the question that is important, because based on that one single question, you have to spend your entire, 5 years of PhD program. So we were fortunate to have very good mentors, who guided us, gave us actually good questions. But finally, after, when we were just finishing the PhDs or entering into new, postdoc stage, we realized that actually it's important to ask good questions in science. So from there onwards, I think from my postdoc, when I just got married, it was 3 years of my PhD, and after that I got married. So I didn't want to, leave my, married life and go and do postdoc abroad. So during my postdoc, I was with my husband in Kerala. So I did my postdoc from India with a DBT fellowship. And, that training in PhD really helped, because during my postdoc, the whole project which I designed, which I defended in DBT, for which I was awarded an independent fellowship, was entirely based on what I thought and what I proposed, the question what I asked. So I'm really grateful to my mentors for that. And even during my postdoc, I was blessed with a very good mentor. So who, guided, and it was a very nice sail through 3 years. And after that, I think I got into a good academic post, so there was no seeing back.
Yeah.
So I think this is how we learn at every phase. So we, learn to ask questions. We learn to do experiments in the lab. We learn to, analyze the results. That's also another important skill that a researcher should have. And then to, translate it.
Yes, that is important.
Yes. So that translation journey, it began when I joined Professor Sareen, because he was the person who was actually working with the patients.
Okay.
So as a basic scientist, we were mostly, until now we were working in the lab. But yes, now taking the lab thing into real world scenario or to the patients. So that I think was mostly I learned from Professor Serene, who is currently my employer also in Institute of Liver and Biliary Sciences. So, working with a clinician, why it is important is because your questions also come from the clinic, and you work towards it like a scientist, and then the solution also you give to the clinic.
Yes.
So
It
it's
becomes
not
real,
that
basically.
yes. So it's like you are actually handling real world problems.
Exactly.
What the clinicians face. So this is something the most important, or I can say the most advantageous niche of ILBS, that we are working in an environment where we are daily interacting with the clinicians.
Yes.
Because they know what their patients are facing. So they give us these challenges. Okay, these are the set of patients we are not able to distinguish. Can you help us with a new diagnostic biomarker? This is the disease we don't understand. This is something which we see new. Can you tell us something about the mechanism? Or and patients with liver, they usually end up in hospitals at end stage, when
Okay.
you don't have any kind of cure or no
Yes.
therapy. So at that stage, nothing is left.
Exactly.
So at that stage, okay, can you give some supportive therapies besides liver transplantation, or at least to bridge that gap until you get a transplanted liver? So that kind of real life situations, real life questions now are coming straight from the clinic.
That is important.
That is important. And so as scientists, I think that kind of niche or microenvironment we are getting in ILBS is something really, really good for us. And then we give our best to solve these problems, and then give solutions which actually should help the patients.
And you have had a beautiful journey, I would say, because as
Yeah,
you
yeah.
mentioned yourself that
Yeah.
you started with a question in the PhD,
Yeah,
and then
yeah.
you were fortunate enough to
Yeah.
actually see the real world impact of that
Yes,
solution
yes.
that you
Yes,
created.
yes.
So that's a wonderful journey you had over there. And as you mentioned, you have been working with the liver biology most of the time and the vascular niche, I would say. So how does incorporating the endothelial and the lymphatic components fundamentally change how we understand the liver regeneration and the pathology behind this?
Yeah. So actually, vascular biology was something which I started during my postdoc. So I worked on cells called as endothelial progenitor cells. So these are cells which actually renew our endothelium.
Yes.
So after that, so most of my projects and most of my studies were designed based on these. So if I talk of liver, so actually during liver regeneration, so I don't know if you know that. So liver is the only internal organ which can regenerate itself even if you take out 70% of it. So if you're left with even 25 to 30% of the liver, it's has the ability to grow back. Original size 100% within 2 to 3 weeks.
Okay, that's great.
Now, the major cells which are responsible, of course, are the mature liver cells, what we call as hepatocytes.
Okay.
But what we are seeing is that these cells need a vascular niche,
Yeah.
right? So, just you heard my story. What is important for me to grow was my niche.
Yes.
So, similar to that, every organ or every cell in the organ needs a niche
to support the growth.
to support the growth. If there's no niche, there's no support.
Exactly.
So this is exactly what my lab is currently doing, to see what kind of signals these endothelial cells are actually giving to the to the regenerating hepatocytes.
to support.
Yes, to support. Then abhi tak whatever work has been done in the liver was in vascular endothelial cells.
Okay.
So just like your blood vessels, there's a parallel circulatory system which is called as the lymphatic vascular system. So it's like a one-way drain, right? So open-ended is blood vessels, but this is like one-way. It's going and then it's getting back into the blood again. So just recently, one of my PhD students, he asked the question whether these lymphatic endothelial cells also support liver regeneration, because they are also a vascular niche, but very less studied because they are quite rare type of cells. It's difficult to study, difficult to isolate, and they are in less percentage in the liver.
Yes.
So, these kind of studies. So, overall, like I said, we are studying these microenvironment signals, factors, how they support liver regeneration or liver cell proliferation.
That's great, actually.
Yeah.
And even the question. So this shows that there can be any question because science is complex,
Yeah.
and once you
Yeah,
question
yeah.
it, it becomes more simpler, and you find
It's complex,
Yes.
but the more easy science you make the science more easy, the more better it is.
Yes, definitely.
The more better it is, because
Yes,
unless I am able to communicate what I am doing, or you'll never understand. So I
Exactly.
don't think it's a good science.
Exactly. So simplifying it and then
Simplifying
communicating it.
is the key to good science. Yes,
Exactly.
very right.
And as you mentioned already, that your team has been developing the liver-specific bioscaffolds
Yeah.
that will be supporting the long-term hepatocyte cultures, right? So, what design principles were very critical to achieve the stability and the function over the period of time?
Yes. So actually, the problem with the hepatocytes is, I just told you, liver is the only organ that
Yes.
regenerates in vivo. But if you take out those cells outside, hepatocytes, they don't survive more than 2 days.
Okay.
So they de-differentiate,
Hmm.
right? They change their hepatocyte morphology to mesenchymal.
Okay.
Mesenchymal.
Oh,
So
mesenchymal
they,
cells.
become fibroblasts, or so they actually lose their functions within
Okay.
2 to 3 days. So we wanted a system now, because we were studying hepatocyte regeneration. Obviously, we had to carry out experiments where we had to culture these cells and then see or treat them with factors to see the effect. But we were not able to get good cells over a long period of time. So for that, actually, we collaborated with few of the scientists or experts who were actually into making scaffolds,
Okay.
bioscaffolds. So I'm not a bioengineer myself, but with a few people in IIT. So what we did, we encapsulated these hepatocytes into alginate scaffolds.
Yeah.
So within the alginate scaffolds, these hepatocytes grow as 3D culture cells.
Yes.
So that helped us to keep this hepatocyte functional for a longer time.
So that kind of became the support that we have been talking about.
Yes, so this 3D system actually allows the hepatocyte to be more functional
Yes.
for a longer time, more stable, and we can use them for testing or something other, any kind of downstream applications. So this thing, and now like you said, what is actually crucial here is that they should not lose their viability.
Okay.
Because you are putting it in encapsulating it in matrigel
Yes.
for 3D, or you're putting it in alginate. So we have to take care that the cells are viable. If there is more cell death, so that kind of thing will not work, whatever you try. So cell viability is important, and of course, if cell viability is there, it is able to survive. So function should come automatically.
Yes. And now that you mention how important the cell viability is important, when we now that you have developed the innovation, right, the question that comes next is how do we scale it up, or how do we see the real world impact? So what kind of problems have you experienced, or do you think are like gaps between doing the scalability or having the reproducibility or repeatability in the domain of developing these organoids or the 3D cell culture?
Yeah, so when it comes to 3D cell culture, so like I said, we grow hepatocytes as spheroids, we grow them
Yes.
as organoids, but we do it on lab scale. So scalability we don't need, because we currently have simple small assays, small experiments which are used for experiments for PhD students. But yes, scalability is definitely a topic which has to be addressed, because currently what we are using is like 24 well or a 96 well plate. But if you have to scale it up for say any kind of regenerative therapy or something, currently we are using it for testing new compounds, or for drug ex vivo modeling or drug testing. But beyond that, so if we need it, we certainly have to scale it up. So maybe we have to use bioreactors or some rotating bioreactors which can scale up the yield.
Yes.
But honestly, like I said, I am not a person who is into bioengineering or this thing, but yes, if I'll need it, then
Yes.
certainly I'll look for it. But currently we are not into
Okay.
this scaling up technologies.
And when it comes to scalability, if, as you mentioned, that you are not from the background of bioengineering, and when you needed something, you approached the IITs for, so this
Exactly.
kind of became a collaboration between academias, right?
Yeah.
So, what are your perspectives, like in general perspectives of the collaboration between academia or academia and industry, because there are many products developed by the industries which will be helpful to do all this research, innovations and everything.
Yes, yes.
What?
Yeah, so if you ask me, my personal, so I have been in collaboration with mostly with the academic people, and uh, again, I'm blessed because I've got really, really good collaborators, and it's always complementary expertise.
Yes.
So, like, we have a kind of expertise in liver cell biology, and those who are bioengineers, like with one of the collaborator, we, bioprinted the liver cells into a scaffold, and the alginate work we did, and then with another collaborator we are now developing some hydrogels. So within the academia, I think complementary expertise is really, really helpful. I cannot do everything, so I need somebody to support what I am thinking. Regarding academia industry, personally, I don't have any experience, but certainly I would like to, see that. And I think this conference 3DCC has been beautifully, curated based on this kind of thought that academia should come in contact with the industry. So I met so many of these industry people who have been developing similar kind of, 3D culture models, cell culture models. And now I think, and the question you asked about scalability, I think probably they will be better answering, or can be helpful to me. So in that way, it will be good to collaborate with some of these industry people and see how we can take our work to next level.
Exactly.
Yes.
And from your experience, how do you think that these advanced systems which are there, the organoids and everything, will be helping to de-risk the late stage drug development? If we already had the conversation regarding this slightly, but if someone has to, some innovation is made in the same domain, and then I want to implement that to the real world instance in patient access. So how difficult it is, and what is the path that takes place during this? What are the steps that actually happens?
Yeah, so I think that was all about the roundtable discussion also we were having just now. So I think these NAMs, or what you call as these new approach methodologies, they will definitely play a very big role in at least shortening the drug discovery pipeline. So currently it's taking years.
Yes.
And even if the drug comes to the market, it is withdrawn based on the results, because it doesn't work. It works well in the small animal, it works well in the large animal, it works well in safety trials, but even then it fails when it comes to, larger masses. So what we currently need to incorporate is human relevant models.
Yes.
So I think that's the whole thing. So it will actually do two things. One thing, it might reduce the animal testing. See, again, the question, if you say it will replace, I don't think, because, physiology point of view, an in vitro organoid will not tell you everything. It will not serve the purpose. If you say I will completely reduce the animals, no. But yes, before going to humans, so it's a human relevant model.
Yes.
It's an alternate to human testing, not to an animal. Might reduce, or you may screen some molecules in these human, reduce that in vivo in the animal. So reduce the number of animals and reduce the failures, the drug failures which are actually happening, because now you have tested in these molecules first in the human relevant models.
Yes.
Now the success again will depend on the robustness of the particular human relevant model, the reproducibility. I have made a model, the drug is testing well in mine. You have made a model, but it is not testing in yours. So then there will be difference. So I need to reproduce it, to validate my results with another model. there has to be some quality standards. Okay, this is tick mark, so it's a good model. So there has to be some guidelines. Now I have tested it in human relevant models. How many animals do I need? Do I need to go to a large animal? So all these things, I think, have to be properly put up into guidelines. And this is all what we were talking about. It will take a few years, but it will come. But certainly, I think these NAMs are the future.
Ma'am, as you mentioned about regulations, right? So, what are your perspectives? How important regulations and policies, or how important they are to accelerate these innovations to come into the real world market? I would say.
Yeah, they are very important.
In
You
what
need,
way?
see, I say it's good enough for my molecule to come into market. The regulators will say no, please go back. So actually, you need a regulator to be saying that okay, this is the correct approach.
Yes.
So that's the whole thing. When today we are making the guidelines of introducing NAMs as an alternative method,
Yes.
we should have regulators on board, because their advice, their perspective is important for us. What do they think?
Yeah.
Do they think that yes, whatever we are thinking, that human relevant models will be enough,
Yes.
or will reduce animal numbers, or I don't need more large animals for testing. What is their opinion? So I think that regulators or those who are into policy making, their thought process, because they are daily dealing with these kind of drug approvals. I'm just confining to my lab, to my molecule, to my model. So probably I may not be having a broader perspective what a regulator might be having.
Yes.
So I will need his advice and his thought to actually say that where does this NAM stand and how we can take it forward.
So we can say that the real world impact solutions depends not only one innovation
No.
but everything around that is
Everything,
there in the
everything.
game.
And if at least we are able to write one or two success stories,
Yes. That is
then
important.
it will change the game.
Yes.
Abhi to we are just talking, okay, it will reduce animal, it will replace. So if we are able to have a molecule which is based on NAM testing and getting into clinic with fewer animals, the journey is short. So that one or two success stories will decide the future.
Exactly.
Yeah.
And looking ahead, as we are talking about success stories, right, taking place, which will definitely help the healthcare to grow and beneficial for the patient and the
Yeah,
Indian
yeah.
population and beyond, obviously. So, over the next 5 or 10 years, a decade, or maybe 2 decades, how do you see liver organoids and the engineered microenvironments which are there are reshaping the drug discovery?
So I think they are going to play a big role. So liver is a very complex system, right?
Yes.
So there are two aspects when we talk of making liver based organoids. So two applications, I would rather say. One is from therapy point of view.
Okay.
Like I told you, for liver transplant, if we are able to have some kind of therapy, and the second is from drug screening, because most of the drugs, they show hepatotoxicity. It's the seat of drug metabolism.
Yes.
So, if you talk of drug metabolism, or for drug screening, these engineered liver microsystems will really be helpful. For in vivo, it will take time, because
Yes.
it's a complex system. I say you just put some organoids in humans and some miracles will happen. I think that will take time. It will happen because people are into bioengineered livers now. I know many groups who are working across the globe, and they are doing really good. But it might take some time. But for your question, which is referring to drug testing, I think these engineered liver systems, liver on chips, so they are mimicking, or they are very close to the in vivo liver, human liver. So I think they will be coming up in a big way and really helping in this drug screening or drug discovery process.
And last but not the least, as we are talking about what is going to happen in the domain of organoids and everything in the coming 5 years or 10 years, when different young researchers and young innovators will be coming with different questions, different innovations that they want to explore. So, what kind of advice do you have for these young innovators and the researchers who will be jumping into this domain now?
See, that's what I told you from the first question you asked.
Yes.
So it's very important to ask a good question. So
Yes.
that the same advice I would like to give to all the young people who are currently into research, that you should improve your skill set, but just don't do blindly whatever your teachers say or mentors say. Please question. At every step you should question, and every failure, we have so many failures as researchers. Every failure you should know why we failed.
Yes.
And then that's the next step you take, so that you don't repeat that mistake again. So I think currently we need to have depth in research.
Yes.
So the students or the young generation should actually read, should think. So they should question themselves, they should criticize their own work. Yes, I was wrong here, I have to improve. Why this is happening, why this is not happening. So the right kind of question will help to dig diamonds. So that's what, the more deep you dive, you dig diamonds. ऐसे तो ऊपर से तो नहीं मिलेगा ना. So you don't have to just stay on the surface and say मैंने PhD कर लिया. It's important to go deep in any kind of research, in any kind of scientific pursuit. You have to dig deep. That's what I can say.
I mean, that's inspirational actually. Before we wrap up the whole conversation over here, I have 3 quick rapid fire questions for you, which is like, I want your very instinctive answers for those questions. So just tell me in one word, describe the future of liver organoids.
Bright.
Wonderful. Biggest misconception about organoid models is?
Misconception.
Yeah, you can take one phrase, not one word.
Okay, they completely mimic a pathology. No,
Okay.
they'll just answer one question which you have asked. It's not possible that a single organoid can do, completely say it's the whole organ. No, not
Okay.
possible.
Now complete this sentence. Human relevant liver models will transform the drug discovery and drug development by?
You want the ear?
Not year, like by doing what? Like by, or maybe you can give me an year also if you can predict that.
I don't know about the ear, but
Okay.
but very soon I can say that. But yes, I think by improving more quality standards
Okay.
and by more reproducible results.
Yes.
So this is, I think, should help.
Okay, it was a wonderful conversation with you, ma'am, because all the thoughts were not only insightful, but they were also very inspirational for the
Yes.
viewers, I would say. Thank you very much.
Thank you.